In pursuit of artificial blood, plasmid expression system for synthesis of hemoglobin and haptoglobin complex in E. coli. I'm Xerxes Paulsetio.
Ian Cox.
Alexander Locke.
And Bori Kim.
The problem we faced was a global shortage of blood donations. There are many issues with donated blood, such as the short shelf life of 42 days at 6 degrees Celsius, and the purity and safety of donated blood, with contaminants such as HIV, hepatitis, and others. Low-income countries have less access to donated blood and less measures to check the quality. Our solution was to create an affordable blood substitute of a hemoglobin-based oxygen carrier, using recombinant DNA technology to create an expression system to synthesize hemoglobin and haptoglobin complex in a bacterial host. Our application is as an artificial blood substitute and in extreme conditions with low levels of oxygen.
Crocodile HB evolved to bind with byproducts of respiration, which allows the crocodile to stay underwater for over an hour in one breath. Using the crocodile HB, we will result in a higher oxygen capacity and more efficient release of oxygen into the tissue. Genes for HB include HBA alpha and HBB. Cell-free HB dissociates into dimers and causes toxicity. HB is known to naturally bind to HB and neutralizes the toxic effects.
So, for our MetAP gene expression plasmid, the first element that we highlight would be the selectable marker, which is the ampicillin resistance gene, and this trait allows cells to survive ampicillin toxicity. Next would be the ORI, which is origin of replication, also known as PVR322 ORI, which starts the process of replication. The T7 promoter initiates the process of transcription, which needs a host with T7 RNA polymerase gene under control of the LAC-UB5. And the expression of T7 is induced by lactose analog IPTG into culture. The T7 terminator, on the other hand, ends the process of transcription. Our spacers are Shine, Delgarno, and 3xGGGS, which starts the protein synthesis process. The LAC-L and the LAC-O produces enzymes which allow the digestion of lactose. ROP increases affinity between two complementary RNAs. The 6xHIS binds to the IMAC column, and the GOI, which are the genes of interest, include the H-beta, H-alpha, haptoglobin, and the MetAP.
The H-alpha, H-beta, HP-MET AP gene expression plasmid has genes H-alpha and H-beta for hemoglobin and HP for haptoglobin. Ampicillin resistance gene, there's also the ampicillin resistance gene for selection. The MET-AP gene is used to cleave methionine residue, an important element of post-translational modification. This plasmid is then transformed into E. coli. The transformed cells are cultured in presence of ampicillin in appropriate medium to select a population of cells containing integrated plasmid. The successfully transformed hosts, grown in media with glucose, IPTG, and hemine, produce the HBHP protein complex. The HBHP protein complex is purified and qualified before given to patients. The complex will be the main component in the drip IV system.
Thank you for listening to our project, and have a good day.